It's so wonderful to see the full hall. Let me welcome and recognize the presence of Professor Charles Wisonge, Professor Ambroise Wonkam and his family and friends in the audience tonight, Professor Raj Ramesa, Professor Liz Lange, Professor Loretta Ferris, academic and past staff members in the audience, students, and members of the public who are joining us tonight. Good evening, Dumelang, Molweni. It's so good to see all of you. Welcome, everyone, to the first Vice-Chancellor's inaugural lecture for 2019. And a very special welcome to our guest of honor, Professor Wonkam. His family must be in the audience. Can I see where you're sitting? Welcome. It's so good to see you. Thank you very much for joining us. To you, the family, I want to say thank you for sharing Professor Wonkam with us. We as UCT benefit from the work of Professor Wonkam, but none of this work would happen without a supportive family. His work is revolutionizing healthcare, but it wouldn't be that if it were not for your support. So we are grateful for the support that you're giving Professor Wonkam. While the practice of giving inaugural lectures is an ancient one, we at UCT feel it is not only fitting, but also relevant to our own lives to give those who have achieved the highest academic rank possible the possibility to share with us that which excites them, their passion and their vision for how life can be made better for others. Professor Wonkam found his calling in healthcare and has dedicated his life to understanding human genetics for the betterment of the public healthcare sector. Having won numerous awards for his contributions, he is a perfect example of how we, at the tip of the African continent, are creating and sharing knowledge with the rest of the world. The title of his inaugural lecture this evening, is Enabling Genetic Medicine. And through this lecture, Professor Wonkam will take us on a journey of his research, which shows that clinical applications of DNA technology could provide immediate benefits for healthcare. The work of Professor Wonkam reminds us of the positive impact researchers from UCT can have on the lives of millions of people. Oftentimes, people out there think that the research that we do is only for ourselves and to advance our own careers. And today, we have this example of the relevance of our research. Relevant impact is the main purpose of all institutions of higher learning. I will now hand over to Professor Charles Wisonge, Director of the South African Cochrane Center, to formally introduce Professor Wonkam.
So, my name is Charles Shea Umaru Wisonge. If you think that my name is long, wait until you see the long list of doctoral degrees that Ambroise has. So it is my pleasure to introduce my friend, my colleague, and my brother from a different mother, Professor Ambroise Wonkam. Ambroise is married to Lily Chungi Oyono Wonkam, here present. They have two sons and a daughter, Ramses Peigu Wonkam, Etam Tamu Wonkam, and Afidi Miko Wonkam. As can be seen from the names of the kids, Professor Wonkam has a passion for ancient Egypt and African civilizations. We call him an Egyptologist for his vast knowledge of the history of ancient Egypt, where the Nubian people from Sudan, with whom he identifies, must have originated. In high school, Ambroise was nicknamed Johannes, a short form for Johannesburg. And this was because of his relatively light skin. Most of them were like me, like white South Africans. Ambroise and I were classmates in medical school at the Faculty of Medicine and Biomedical Sciences at the University of Yaoundé from October 2008 to February 1995. Don't start calculating my age. This was the only medical school in Cameroon at the time, and Ambroise and I were among the handful of students who were admitted directly from high school to medical school. Must have been very clever. The rest had spent a number of years at the Faculty of Science of the University of Yaoundé 1. Ambroise was, I think, the youngest student in the class, but academically, he was the top for all the 3 academic terms in year 1. He was the top of the class. But I can happily note that when we graduated from medical school in 1995, Ambroise and I had the same mark. To 2 decimal places. When the marks for clinicals and doctoral degree, and doctoral thesis, Ambroise was not only tough in class, he was the first editor-in-chief of Canal Medical, which was the scientific journal of medical students of the Faculty of Medicine and Biomedical Sciences at the University of Yaoundé 1. After medical training at the University of Yaoundé 1, Professor Wonkam completed a Doctor of Medical Sciences in Cell Biology at the University of Geneva in Switzerland, and a PhD in Human Genetics at UCT, and I think he's here. Professor Wonkam also trained as a medical geneticist at a highly reputable genetics department in Geneva in Switzerland. He subsequently practiced medical genetics in both European and African contexts. Professor Wonkam is a professor of medical genetics here at UCT, and he's the director of GeneMAP. He is also the deputy dean for research here at the UCT Faculty of Health Sciences. I am actually aware that Ambroise at some point used his personal money to pay for research for students. And I hope that in his new position, students and supervisors should never have to do that. Professor Wonkam has published more than 130 peer-reviewed publications in molecular, clinical, educational, and ethical aspects of medical and human genetics. His publications are highly cited. I checked today, and his publications have been cited more than 1,638 times. And 25 of his publications have been cited at least 25 times. He has an H index of 25. Professor Wonkam's research focuses on 1, psychological burden and genomics modifiers of sickle cell disease, 2, genetics of hearing loss, and ethical and educational issues in human genetics in Africa. Over the past 5 years, Professor Wonkam has been very productive in bringing money to this institution. He has successfully led an NIH-funded sickle cell disease project and was a co-applicant on the Wellcome Trust Delta's grant to develop capacity in human genetics in Africa. He has recently been granted $3.7 million from the NIH to establish a sickle cell data coordinating center, SADC, to develop various studies in Tanzania, Nigeria, and Ghana. In addition, under the new round of H3 Africa Consortium, Professor Wonkam was awarded 3 grants to support the Hearing Impairment Genetic Studies in Africa, HI Genes Africa project from the NIH, $1.25 million. And ISA Wellcome Trust, $2.07 million. And another collaborative center grant from the NIH, $2.5 million. I will have to stop calling these figures now. To support the study of incidental findings in genetic research in Africa. Professor Wonkam was awarded the 2003 Denver Pinard Prize for the best thesis from the Faculty of Medicine, University of Geneva in Switzerland, and won the very competitive Clinical Genetics Society International Award for 2014 from the British Society of Genetic Medicine. He is the president of the Pan-African Society of Human Genetics, a member of the steering committee of H3 Africa Consortium, a board member of the International Federation of Human Genetics Societies, a council member of the Human Genome Organization, and a steering committee member of the Global Genetics Medicine Collaborative. With 3 doctoral degrees under his belt, I would imagine that if he was working for a German institution, he would be referred to as Professor Dr. Ambroise Wonkam. I don't know about you, but this sounds like music to my ears. And I am certain that the melody will be highly present in his inaugural lecture, Enabling Genetic Medicine for African Populations. Thank you.
So, bonjour, bon après-midi. I have to complete the language from the VC because he didn't say French. It's really, really a privilege to be here to share some part of my journey with you. I will start to dedicate this lecture to my father, my late father that passed away about 5 months ago. The reason being that he's been a very good father to us. He's been my role model for a very, very long time and still living by his principle up to now. I have a thought for my mom. She lived with my father for 55 years. And now she will have to live a little bit, a few years, I hope, alone. I have a thought today for some other people that I don't even know, anonyms. When I was 18 years old, I was about to prepare to go to France for study, and I have my first passport. On my passport, it was written that you can travel to all the countries in the world but South Africa. And this was in 1988, because in South Africa, of course, people like me, African people, were not necessarily well treated. So if I'm sitting here today, it's because of many anonyms that sacrificed their lives to make this place a better place for everybody. I would like to thank a few people that allow me to be here. First, Professor Raj Ramsar and late Bongani Maosi. I met these two gentlemen in 2006 in Addis Ababa in an international meeting. And at that time, they tried to convince me that there was no space for me to live in Europe and that I will have a better ground here. And they knocked on my door for subsequent 4 years until I landed down here. And I think it was a really good decision to move here. And I would like to thank Mariam Jacobs, because she was dean at the time. And I must say she went out of her way to get me some of the administrative hurdles on the site. It's difficult to stay in an environment that is not professionally fertile if you don't have a good relation with your colleague. So it's my pleasure to thank Karen Figan, with whom I've shared the medical genetics for many, many years now, and my friends and brother, Colette Dandara, for your faithful collaboration over this year. Of course, I would like to thank all of you here today because I'm very surprised with the numbers, because I thought this is a very niche area and that not many people would necessarily fetch up. But I would thank especially a very special person to me, my wife, Lily. She got 4 major characteristics that is rare to find in a single person. She's beautiful, she's elegant, she's intelligent, and she got a very strong personality. And most importantly, she has been a very, very supporting wife and partner and an excellent mother for our 3 children. And I have to thank her especially because I know she sacrificed a lot to follow me around the world, putting in bracket her own perspective, a career perspective. You are the best. Of course, it's difficult to have an academic journey without a strong support. As Charles said, we have the means. We just need to work to do what we want to do. And we are strongly supported by the NIH and the Wellcome Trust specifically. Today, I would like to convince you that we need to invest in genetics to improve medicine and specific medicine in Africa. But before I get to that, let's start a little bit with the history. Genetics, of course, have always existed since the beings exist. But modern notion of genetics was put to us by a monk called Mendel in 1865 when he described the law of heredity. One of the most important moments is when these 2 gentlemen, Watson and Crick, show us the structure of DNA. I think an anecdote is that Watson and Crick spent a lot of time in the bar of the university, but not necessarily in their lab. So it might be good to spend time in the bar. And one element that is also important is Chijo and Levan that describe that we have 46 chromosomes. I'll come back to that a little bit later. Chijo was a Japanese student of Levan. Levan was a Spanish scientist. And in fact, Chijo is the one that really described the 46. When he came back to the boss and said there is 46, the boss went and looked and said, no, it's 48. So there was a challenge of the student to the boss, and the student left, and he wrote the paper and said it's 46, and eventually, but he couldn't comply. But the most important element, really the most important revolution that happened a few years ago, the Human Genome Project that was declared completed in 2003 by Bill Clinton. This project was designed by the United States of America, and because they started the project, they didn't know how we're going to read this thing. And they get inspired by Africa. If you look at the right-hand side here, many of you may know this stone, Rosetta Stone, that is at the British Museum now. This stone was discovered and decrypted by my uncle from France, Jean-François, you don't know yet, Jean-François Champollion. Jean-François Champollion, when he discovered the stone, on the stone there are 3 scripts. One is in Egyptian, the second is in Greek and Roman, the third one is in Demotic, which was kind of Egyptian for the lay public. He knew very well how to read the Roman and a little bit the Demotic, and he was able now to do the comparison and decrypt it. He wrote a letter to his brother. The brother is called Champollion-Figeac, and say, ceci, c'est le résultat des comparaisons. So when the scientists that started the Human Genome Project, they just inspired themselves from this, and they were going to compare many, many areas of the genome to be able to read it. The first level of comparison was the comparison of our genome with our genome. The second, our genome with the other species. The third, the comparison with us and our neighbor. And the last, the comparison with cell state. For example, a cancer cell in the same organ versus a cell that is not cancerous. What have we learned? We learned that from this project that our genome is organized like a big library of life with 46 shelves. The geneticists call the shelves chromosomes. And with 20,000 books, the geneticists will call the books genes. We know that, in fact, part of our genome that is responsible for our function is a very, very little proportion, only 1 or 2% that constitute what we call genes. And by comparing our genome to the other species, we are very closer to some of the species more than we thought. For example, our best friend should be mice. We always think it's dogs, right? But no, they're not dogs. Mice are our best friend. We have the very same number of genes, 20,000. And we have 95% function, same function like mice. Someone will say, I thought it was chimp. Yes, it's okay. Chimp, we have 98% similarity. But I will tell you mice beat chimps on this friendship stuff. If we have this room, we can grow a lot of mice. And for this whole university, this only on this space. But this will not be enough for one chimp, because he's going to beat all of us and we're going to run away. You need much more space. Pregnancy in a chimp is 9 months, exactly like us. At the end, you have one baby chimp. Pregnancy in mice is exactly 19 days. At the end, you have tens of them. Trust me, I spent 3 years of my life working on mice. So, and if you have the very same function, that means that it's the best experimental animal that we can use. And why is that we are clever than mice? Just because we are able to do 3 times more function, more protein. For example, you give a pot of rice to the mice, you're going to just eat the rice raw. You give it a pot of rice to any lady here, you may have 3 or 4 types of recipe, right? And with different tastes. And another very important element here that the human genome shows us is that we are all Africans. We are all Africans is important. And is this acceptable for everyone? No, at the beginning it was difficult. For a very long time, archaeology used just archaeological type of study to define where the human come from. And for example, this Englishman called Charles Dawson in 1912 discovered this skull that is called the Piltdown Man, that was supposed most ancient skull than the skull found in Africa. But fortunately, another Englishman called David Waterston showed that there was a forgery here. The head of the skull was from a chimp that effectively was older than humans, but the lower section was from a modern human. It was astuciously made up, put into the ground, and discovered. But with the Human Genome Project, we know with a perfect sense that Eve, the biblical Eve, was African. Using mitochondrial DNA, we can know that DNA from African population are linked directly to oldest mitochondrial DNA, and which is not the case for the mitochondrial DNA from other population. And everybody migrate out of Africa. And in fact, migration is part of human journey. People move from Africa to Europe, and then move from Europe to America, and now move from America to the moon. So, and again from the moon, they will have to move from somewhere else. So, and another element that Human Genome Project show us, not only we come all from Africa, but we are all very similar genetically. 99.9% similar, as despite the way we could look, the way we could speak, the way we could cry, or the way we could laugh. But are we that similar? What is so specific in this toolkit with albinism? What is so specific in this individual with blood clot disorder? And in this man with hypercholesterolemia, on this other kid with Down syndrome? What they all have in common is genetic variations, all of them. They have one specific changes in their library that makes them different. That changes may be a change that happened at the level of the shelves, what a geneticists would call chromosome. For example, for these two kids, they have 3 chromosomes, number 21, 3 shelves, number 29, instead of 2. These changes may be just that in one specific book, you may have some repetition of letters and called short sequence repeat. And this short sequence repeat allow us, for example, to do filiation testing, to do if someone is the brother of some other one, because you should have the same type of repeat. These changes also may be just a single letter changes, what we call single nucleotide polymorphism. And from you to you, from Ramses to Etam, every 1,300 letters, there is a change that differentiates you. Not really, you share 50% of your gene. You are my sons. But some of the changes can help us resist to some diseases. For example, in sub-Saharan Africa, where most patients with sickle cell disease live, is just because having the mutation allow us historically to resist to malaria, and it become extremely frequent in sub-Saharan Africa. And there is one specific change I would like to mention here. If you have a deletion in this gene, that is the receptor on white blood cell, a double deletion from the chromosome from your dad and the other one from your mom, you will be HIV resistant. And this was the basis that these Chinese scientists this year report for the very first time a first baby that was gene edited for CCY5, and she will never have, she's a baby girl, she will never have HIV. Of course, there is a lot of ethical contestation on what happened, but it happens. It's the proof of concept that we are able to change later in a baby before he's born, and he will be born different with something resistant or not. The Human Genome Project have allowed us to have every single day new disease genes, every single day. So the number of new disease genes is exponential. That have allowed to generate another type of genetic specialty, which is called medical genetics. It's always very hard for me to explain what I do. Because you all know, I know everybody, so okay, today I want to go and see my GP. I want to go and see my gynae or my urologist. But I've never seen someone saying, I'm going to see my geneticist. So one of these days, my little girl asked me, what do you do, Daddy? So I explained I was a geneticist. I said, what a geneticist does, okay? Let's imagine these letters. A, T, C, G. These are the letters of our genomic material. And imagine these 4 images, and let's try to connect it. In fact, what I do every single day at matchmake. You understand what I'm trying to say. So what geneticists do, we try to look for variation or later changes in the human genome that may appear like this on a sequencer, try to make sense of it, and communicate that to our patient. And this may mean changes in the shelves. This may mean changes in the books. For example, here, Down syndrome, 3 shelf. We saw this picture already. And here, a specific 1 letter change here in this specific book that is called FGFR3 gene that is located on this specific shelves, which is number 3, will lead you to a set condition that is called clinically achondroplasia. As Charles said, I spend all my time, or I used to spend a lot of time reading history books. And I realized that, in fact, achondroplasia was already well known in old Egyptian. And in fact, this man was a priest, and he was a counselor to the pharaoh. This is an important piece of information because it means that 1,000 years ago, we had a better tolerance to difference than we have today. And what does this help us when we have those information? This can help us improve our diagnostic. This can help us look for predisposition for disease. This can help us design new genes that are related to drugs efficiency. This is helping us now to improve the diagnostic of HIV, HPV, TB, or malaria, and this helps us a lot on forensic genetics. Okay, let's move now to when it's come to Africa. Is it easy to implement? Is it important for the continent? I will start to show what are the problems. The first type of problem we have on the continent is investment in innovation and wealth. Despite the fact that we are 15% of the world population, only 5% of world gross domestic product belongs to Africa. So we could be modernly called relatively poor. It's not necessarily true, because we are more stolen than poor. And we have 1.3 of the global investment research on innovation. That means that we don't invest a lot on research. The second problem we have is knowledge. Once again, I started by saying that when I explain to someone that I'm a medical geneticist, they will start by saying, what research do you do? I say, no, I see patients. I don't do just research, I also see patients. So the knowledge of genetics is very low, even among medical professionals. Among the published, the knowledge is also relatively worse. For example, in this specific study we are doing now, in this village where there is this condition called Fragile X, it's been known that this was due to a curse that was thrown at the chief, and to have all his son mad, because this condition is associated to mental retardation. But in reality, it's a genetic condition. The other problem is that there is a gold rush now in Africa. Everybody won't have part of Africa DNA. But if we look at the type of study that have been performed and the publication that come out of it, very little African authors are included in the publication and very little African institution. That means that the researcher take the sample, fly out, what is called helicopter research. And most importantly, if we look at the topic of research, most of the research internationally focus on human genetic variation because it's quite sexy. You publish in Nature, in Science, and so on. But very few will be interested in doing specific disease conditions that are relevant to the continent. Of course, we are not going to expect anyone to do our work for ourselves. And most importantly, but this is danger for everyone, not just for African. This is danger for the whole world genomics. If we look at big studies that are performed on the African continent or globally, the genomic data that are available internationally are 96% European about 2009, and 2 years ago it was 81% European. Less than 5% of genomic data in the public domain are African data. Why is it dangerous? Because remember we say the oldest population is African population. That means that we have 10 times more variation than any other population at least. That means that without studying those variations, we are having a missing link for many genetic association study. And this is a critical element. I'll come back to that a little bit later. But these have been changing with major initiatives like Malaria Gene Project, H3 Africa, the Wellcome Trust and the African Partnership for Development, the South African Genome Project to be local. And having this massive investment have allowed more African scientists to get involved in genomic research in a purposeful way. And if we were to perform the same study, author versus topic versus publication, today we will realize that the trend have changed completely. Of course, this slide is extremely biased toward my work, but that's normal, it's my day, right? So, and with this interest, we are now also building more and more capacity according to the principle of my friend Bongani Maosi, live as you rise. And we believe that in the next 10 years, we should be able to have more and more African scholars that are doing genetics based on the African continent, and that will produce enough of data that are relevant to the continent. I will now start to illustrate this by three stories. The first story, I call it the tragedy of the common, which is the tragedy of sickle cell disease. The reason why I have to start with sickle cell disease is not because it's my research interest, it's because it's the first molecular disease of humans. It's the very first condition where someone shown that you change a single letter, you have a potentially lethal condition. I have to speak to this also because I grew up professionally with a very strong tradition on very, very high profile people in sickle cell disease. Started with the great grand mentor here, Pauline, that won 2 Nobel Prizes for his study on sickle cell disease. Also with Greg, in fact, I met Greg for the very first time in October last year. And he trained my direct mentor, Antonorakis, that described the haplotype of sickle cell disease. Of course, there is a question mark, you see, because I don't know exactly what the history will remember from what I'm doing, right? But I'm working very hard for it. Sickle cell disease is a disease that evolved on the African continent probably at least 7,000 years ago, and get fixed in our population about 5,000 years ago because of malaria. And now every single year we have 300,000 newborns that have this condition. And what is sickle cell disease? This is a mutation that allow hemoglobin, that is the main protein of red blood cell, to become relatively rigid, allowing the red blood cell to change from a round shape to a banana shape. And these cells are relatively sticky, so they will tend to stuck on red blood on blood vessel. And if you don't have oxygen, because the role of red blood cells is to bring oxygen, you have pain, you have chronic pain, you have organ failure, you have possible sensitivity to infection. In Africa, he estimated that if someone has sickle cell disease without treatment, they will die before year 5, half of the kids. But if you're out of Africa, you will live a little bit longer, but data from the United States show that for the past 30 years, the mortality for adult sickle cell disease have not changed, despite the fact that this condition was described more than 100 years ago. And there is no treatment. There is no efficient treatment for sickle cell disease. So it's definitively a very common but neglected disease condition. Why do they die? They die because the disease allows the kidney to fail. The disease allows the brain to have stroke. The disease allows the heart to stop working. How can genetics help this specific condition? We think genetics can help at the level of primary prevention. And this slide is absolutely marvelous. No, not the slide, it's the name of that baby. And this baby is the first baby that was born after genetic diagnosis before, but she was born in Cameroon, and we introduced diagnostic in Cameroon. And my wife may remember very well how hard this was, because I have to establish the lab in my kitchen. The reason being that at the time, there was no lab, and no one even knew what I was doing. And there was nothing, so I had to establish it at home. So this very first diagnostic was done in our house. And we established the very same here in Cape Town in 2010. But if you diagnose a fetus before birth, that means that it's open some other ethical question, because this pregnancy, if affected, might be stopped. So we thought that that was not necessarily the right direction to go. Maybe in the right direction for research is to look for factors that can help save the patient or cure the patient easily. We know that some patients will live up to 70 years without any problem. For example, sickle cell patients that are living in the Middle East or in India. But sickle cell patients in Africa will die very early because they don't have a very high level of fetal hemoglobin. And this is controlled by genetic factor. So one of the things that we have spent a lot of time doing is to look for those factors that control, for example, for this study by Amy, a renal dysfunction, for those factors that control stroke in sickle cell disease. And our hope is that in future, if we have a child that is born of sickle cell disease, by looking for all the variation in many, many genes, we may add the mathematician to design a model that may be able to predict that this child will have stroke. And if child will have stroke in the future, we start blood transfusion immediately. This is not fiction. This is already happening. And for now, there are even transcriptomic studies that can predict if a child will die early or not. And lastly, genetics can help for treatment of sickle cell disease. Once again, the reason is based on the fact that if you are a patient with high level of fetal hemoglobin, you live longer. If you have low level, you live a little less. But fetal hemoglobin is all controlled by some genetic factor. And if you manipulate some of these factors in a mice model that was initially sickle cell disease with banana cell here, if you remove this specific gene here, the mice is cured from sickle cell disease. And this was the basis for the very first gene therapy that was reported last year for sickle cell disease. Another gene therapy cases were reported last week for sickle cell disease. So clearly, we have 300,000 new baby, we should be starting investing in this. And it's important to say that everything we try to do, we try to connect with the community. And last year, during the World Sickle Cell Disease Day, we launched the South African Sickle Cell Disease Support Group with the help of doctors from Red Cross and from Grutus Cure, and that will allow us to improve our inroad and collaboration with patient support group. The second story I would like to tell tonight is the tragedy of rare diseases, and the focus will be disorder of sex development. And many years back, when we tried to establish a genetic service in Cameroon, we realized that there was a lot of patients that have this condition with urogenital malformation. For example, this is a number we had the very first day of the consultation. And because we were very poorly equipped for medical treatment and for diagnostic, I called a few friends of mine, Pierre-Yves from Lyon and this friend from Geneva here, that was the former head of pediatric surgery in Geneva. And we were able, through this program that is still there, the program now is fully funded by the Cameroonian government now. The program has been there for more than 12 years now. We have been able to map, for example, for some genetic condition, the map of the country for the type of mutation that we have per region and so on. And we were able also to provide some solution for some patients. And this case is very interesting. This young boy was seen by us. He was 14 at the time. He was self-identified as a boy. But by the time he came to the consultation, he had a breast development. He had a phallus, a penis, but when we perform a fibroscopy, we realized that on one side he has ovary, on the other side he has a testis. Of course, his life was really miserable because every single morning he had to tie his chest so that his peer would not notice that there was something unusual. So after the appropriate diagnostic, of course, based on genetics, after the appropriate counseling, after the appropriate legal step, we were able to provide the appropriate treatment for him medically and also surgically. This single case means that in many, many areas of the world, in many, many villages in Africa, there are people that are suffering in silence in the corridor of desperation. They don't know where to go. Even if we needed to have only a single genetic service, we need a single genetic service for each country because rare disease are not just disease for the rich. This was also a case where you can combine a relatively complex discipline together. It's like saying that beauty and beast live together, right? Genetic and surgery, because we needed the two, okay? Beauty is genetic, of course. And this program gave out a lot of experience, and I think this is probably one of the most influential papers we ever write. And in 2010, we wrote this piece of paper that recommended very strongly that the International Federation cannot use genetic for gender testing because we are not genetically literate enough to do it. And I sent the paper, I think it's a paper that was written in a week. We sent it, and 3 days later, we see what accepted because the editor was so in urgence to publish it. There's a lot of spelling mistake in this paper, in fact, when I read it retrospectively. And a couple of weeks later, we got a nice letter from the American College of Genetic Counselors telling us that, in fact, people that argue against using genetic for gender testing use our paper. And what is funny is that they are still looking for other ways of using hormonology to single human beings. But in the paper, I remember very well, we say, if the International Olympic and Sport Regulation Body maintain gender verification in the future, even an etiological or functional classification will not necessarily allow a fairer basis for competition. What is the range? What are we going to say now that Michael Phelps will not swim again because his span is very, very large, right? And he has a genetic condition that allows him to have a lifespan. Should we exclude him from swimming? That would be good for our South African, right? So, because he would definitely won all the medal all the time. The last story I would like to speak to is called Unknown in the Exomes, and the model I will use will be genetic of hearing impairment. Hearing impairment is called the silent epidemy. And hearing impairment on the African continent have the highest frequency than anywhere or in the world. In South Africa, for example, 6 kids out of 1,000 are born with hearing impairment. In Nigeria, it's about 7 out of 1,000. And half of the kids that will be born with hearing loss will be due to a genetic factor. And when it's come to genetic, some of the kids born with hearing loss will have hearing loss with something else. I'm sure that you have seen in the street at least one, someone with forelock, the white hair in the middle here, right? And in that condition, people can also have very blue, striking blue eyes, and sometimes very different. Karen will call this heterochromia. Sometimes it's both sides. And it's very impressive in someone with black skin. But if you see someone in the street that have a white forelocks, ask the person to go for hearing testing. But not send the person to me, right? So it will be much better. So, but most patients will have non-syndromic. Non-syndromic means hearing impairment alone and nothing else. And if you are working in Europe, if you are working in China, most of the genes that are responsible for non-syndromic hearing impairment is this book here called GGB2. That will be account for 50% of cases. And when we started this study here in Cape Town, we were very excited. We gave the project to Jason, and Jason came back and said there was no mutation out of the 200 samples from both Cameroon and South Africa. Initially, I thought you were suspicious because how is that there is no mutation? We throw in the sample without telling the student a specific control that we knew had the mutation. He came out with this mutation. So clearly, it wasn't the problem. And we wrote to a colleague, Richard Smith, and say, I'm not having GGB mutation in my sample. He said, no, that cannot be possible. And I said, do you have some sample from African American? He said, yes. Can you check? He checked and get back to me and said, there is none. And then we started, that's how the research started somehow. In fact, if you look at the other genes that are responsible for hearing loss, there are about 100, and test for all of them, like 180 genes. In South Africans and Nigerians, the detection rate is very low, 4%. And if you perform the very same in African American, the detection rate once again is still low, 26%, compared, for example, to 70% in American of Middle Eastern ancestry. And based on that, we used recently a family, and we are still doing it now, looking for that was negative for those testing, and use a technical whole exome sequencing, Whole exome sequencing means the 20,000 books, we read them all and look for the books that have some mutation that may be linked to hearing impairment. And we were able to found this novel mutation. And to prove that it was really a novel mutation, we performed some kind of test. This was done in Ed Sturrock lab here. That shows that there was an instability of the protein. And also we looked at the expression of cell line when the protein was not there and the protein was not appropriately produced and not appropriately addressed. Unfortunately for us to confirm it, there is a mice model that doesn't have this mutation, and that mice model is deaf. So the study is completed now. We just need to wrap the story and publish. And it's critical that every single place where we are doing the study, we engage with the community. For example, this is a meeting with all the head of the school for the deaf in Cameroon. This is a meeting with family in Limpopo. This is a meeting with a specific school for the deaf in Ghana. And this study is also happening now in Mali. It will be happening in Sudan if they release our friend Muntasa. And it's happening in Rwanda. Why this is important? This is important because the panel for diagnostic that are available on the market now are not appropriate for people of African ancestry at all. Detection rate extremely low. So we should be able, at the end of this study, to provide a diagnostic panel that is appropriate, that is relevant, that can be used everywhere else in Africa and in African in a diaspora. Why is this important? This also is important because, remember I told you that we have very few representation of variation of African population in databases. This dictionary here is called EXACT, where it's a dictionary where every time we have a mistake in a book, a variant in a gene, we compare the variant to the dictionary. Is this variant exist in the dictionary as a mutation or not? But if the dictionary doesn't capture all the variation, that means that if you are doing this research in Africa, you may have a lot of false positive or false negative. So what we will do is to contribute to improve this dictionary. This is not charity business. It's not African plaguing poverty again. No, this is the science that have to be done for everybody. The typical example came from the literature a few years ago, where there was a specific variation that was labeled pathogenic. And many patients have been given that diagnostic. Some of them had even have an intracardiac device until a clever scientist say, let me look at this variation in all the population. And when they included African American population, 30% of the American had the variation. So it was definitively not a mutation. So they have to call back some of the patients, and that was a terrible tragedy for that center. So having diversity in exact databases is good for everyone. And this slide borrowed without permission to this very clever man called Victor McKusick, that is considered as the father of modern genetic medicine. And I was fortunate to be in one of his lectures in 2003 in Bar Harbor. And this was his expectation of how the genetic would evolve. He thought that in 2020, that means next year, gene-based drug for diabetes, hypertension, and cancer will be available. I think there are many drug-based therapy for cancer, but not really yet for diabetes, hypertension. But I guess we still have 1 year to work, right? But one thing that was very important for him is he thought that the worldwide inequity in access to technology will contribute to international tension. But what he did not anticipate is that that tension will not start elsewhere than in the US. A couple of years back, there was this company, the company still exists, called Myriad Genetics, that patented a genes. I think you probably heard about Angelina Jolie's story about one specific gene that is responsible for breast cancer, BRCA1. So this company patented that gene. That means that if you are doing a diagnostic, you have to pay a royalty to the company. At some point, there was a revolt, first of all, from the patient association, secondly, from the American College of Medical Geneticists, and then the American Society of Human Genetics, and they brought the case to court, and Myriad Genetics lost the case. And this was the Supreme Court of the United States that decided that you cannot patent part of a human genome. But what Myriad did, Myriad just moved the patent to Australia. Why am I telling this story? I'm telling this story because genetic technology should be a priority for each nation. Let's imagine that someone, Myriad Genetics, discovers something that is critical for our population. That means that we will have to pay for the diagnostic. But if we discover it here at UCT, we don't have to pay for it. We're going to just put it to NHLS, and the NHLS will do it for free for every single body. In fact, if we look at this picture that was provided by Francis Collins, director of the NIH, he thought at the time that, and is still the case, that Human Genome Project is here. We still need to build all these layers with all these pillars. And this big house, there is a key to get in. My view is that we are still outside of the house. As African scientists, we need to have the key. And can we do it? Yes, we can. We need to inspire ourselves from our forefather. I told you I like Egypt, right? So this is the real father of modern medicine that wrote a lot of textbooks, detailed textbooks of medical procedure and diagnostic that was even used by Hippocrates and belong modernly to Hippocrates, in fact. And if we inspire ourselves for this, if we work hard enough, I think international community will trust us. And if they trust us enough with the national community, we will have and the means and the will to get it right. And my hope, my expectation, what I've been working for, what I hope to work for the next 20 years, is that maybe when I give this lecture again, Africa will be visible in the map of innovation and wealth creation. And I would like to share this dream with all the young people that are in this room.
Good evening, colleagues, Vice-Chancellor, the Wonkam family, and friends. We don't gather often enough, and that's probably the first thing that I'd like to say thank you for. The presentation of an inaugural lecture is not a tradition throughout the world. In the USA, the title of professor is loosely used, and generally it is associated with the noble profession of teaching. So high school teachers, most of you will be familiar in the United States, are called professor. Not here. Perhaps the way we use it, it is part of the British and European tradition, and I know this is not popular in the age of decolonization. And it speaks to the deep respect by academics to a colleague who has risen academically to hold the position of professor, the highest academic rank at a university. In terms of my job tonight, it is perhaps one of the easiest and most pleasurable things for me to do, that is to deliver this vote of thanks. I first met Ambroise at a meeting of the African Society in another African country, and you heard it was Addis, and heard of his training in medical genetics in Geneva, as well as his being involved in research. And I could not help but urge him to get back to the African continent, and I remember that conversation with Bongani. When I next saw Ambroise later in Egypt, I was thrilled to hear he was indeed back in Cameroon, where he had been for the last year or two. However, he looked crestfallen and disheartened by the lack of resources and support he was getting. I hope I'm quoting you correctly, Ambroise. For the lack of support he was getting, and he was about to respond to the ever-present call to return to Europe. Perhaps something you may not know about is this thing called alignment of the stars. It was at the Cairo meeting of the African Society of Human Genetics, where I, as part of the executive of the African Society for Human Genetics, presented a vision that the executive was developing regarding genomics and human health on the African continent in an Africa-wide approach. Things seemed to the executive of the African Society to be coming together, but obviously Ambroise at that stage was not exposed to it, so I remember taking you around the corner and beating you over the head about this. So, at the Cairo meeting, where we had presented this, things seemed to be coming together. Part of this ambitious, and I stress, unfunded program was trying to repatriate trained colleagues back to Africa. So just extending the astrological metaphor, where I talked about things lining up in the sky, you obviously won't realize this, but Ambroise and I were born on exactly the same date, the 8th of December. Just happens to be a few years apart, obviously. We were in Egypt in front of a hotel called Ramses, and we were having a conversation, and Ambroise told me his son's name was Ramses. I said, things have to move in this direction. So with absolutely no resources at my disposal, I urged Ambroise to come down to Cape Town, i.e. instead of going back to Europe. And that was if only to remain on African soil. Now that was an unbelievable gamble. I did not have a position, and I've never been at UCT when it was not a time of austerity. Suffice it to say that for once, the arduous process of applying for accreditation of his qualifications through the Health Professions Council of South Africa and recognition as a medical geneticist, which would have put off even the strongest of Africans, has provided me with enough time working with Bongani Mayosi, who was the head of medicine at that stage, and Professor Marion Jacobs, who was the dean. The Department of Medicine is where our medical genetics unit is located, to put resources together to support a post for Ambroise. So again, this speaks to the old axiom of where there's a will, there's a way. It was absolutely the right thing to do. You have heard in the introduction from Professor Vice-Chancellor the glowing comments made about Ambroise's professionalism and his dedication to the cause of medicine and sciences in Africa for its people. Ambroise's coming to Cape Town had not been easy. Moving with a young family is often fraught with tensions regarding spousal careers. Lily, his lovely wife, I stress Ambroise, his lovely wife, has been fantastic in holding the family together while herself developing her career and being registered for a higher degree. Lily, to you and your 3 beautiful children, the 2 boys, Ramses and Etam. I had a beautiful presentation showing you the kids when they were little and you were at home. And this gorgeous little princess, I understand you call her that, Ambroise, Afidi. Thank you very much for being flexible. And I'd like to take this opportunity to wish you everything of the future, everything of the best for a remarkable career which beckons you in the future. Obviously, as a good geneticist and as a good African, we thank our ancestors. In this regard, it's a pity, Ambroise, your parents did not witness your inaugural lecture. However, I'm sure Mom from Cameroon and Dad, who recently passed away, are watching over you. I acknowledge their contribution to you being here with us. Professor Wonkam, your inaugural lecture has given us great illumination, amusement, and pleasure today. On behalf of this audience, and indeed all those who could not be here, I thank you for the lecture, which more than celebrating your achievements, illustrates the great promise for the future. There is little question that your continued tenure of the chair will be of great benefit and advantage to the university, including our scholarly community of students and colleagues. More importantly, we share a very powerful vision for genomics contributing to human health here on the African continent. May your tenure be auspicious, may it be significant for the future, and may it be attended by every good fortune. Lastly, in your name, I honor Bongani Mayosi, whose vision for this faculty and its relevance to Africa we remain committed to. Ladies and gentlemen, thank you. Siyabonga Nkosi.
On behalf of Council, Senate, and the entire UCT community, we congratulate you on rising to this level of professorial level. Congratulations on your achievements. Your lecture was very inspiring. You said it's a niche field, but actually as we were talking, I thought, wow, more people need to hear this. Thank you very much for the contribution that you've made already and what you will continue to make to not only UCT, but to South Africa and the region, and also to the transformation of the discipline, but also of our institution. We are very blessed to have you in our midst. Thank you for choosing us. Colleagues, thank you very much for joining us in this lecture. In a moment, the procession will join me here and we will leave the hall. Thank you.